Hi, everybody. This is Dr. Ed Park, and we're joined by Dr. Michael Fossel in what's going to be a brief discussion of his exciting new book, beautiful book, The Telomerase Revolution, just out this year. And we're going to talk telomeres and answer some questions. So let's get started. Dr. Fossel earned his MD and his PhD from Stanford. He lectured in neurobiology and research, and he was a professor for 27 years at Michigan State in the School of Medicine. He's been at the nexus of a lot of interesting intellectual movements. He's been the executive director of the American Aging Association, and he was the founding editor of what's now called Rejuvenation Research. I think it was a journal of anti-aging medicine. He wrote in 1996 Reversing Human Aging, and at that time, I guess it wasn't me that came up with the telomere theory of aging. It was at least partially Mike. And then again, every decade he seems to reiterate this message, and he's just waiting for the world to catch up. He wrote a clinical textbook, Cells, Aging, Human Disease, which was really the standard, and university course on aging came out of that as well in 2004. As a result, he's been asked to lecture at the Smithsonian, National Institutes of Health, and is a frequent guest on major outlets for news such as NPR, Fox, CNN, and the BBC. The book, which I have read cover to cover, is a fantastic read. In fact, it was named 2015 Best Science Books by the Wall Street Journal. This is something like the review I'm going to put on Amazon and Barnes and Nobles. I would say it is scholarly yet accessible. It provides a logical and very optimistic view of the history and of the future of telomerase activation medicine. I like it particularly as people who watch my podcast know I love analogies. It's got some great illustrations, and it provides a really clear roadmap of what he believes and what he disbelieves. And there's a lot of faddism in anti-aging, and he addresses that quite roundly. There are 8 chapters. The first chapter is Theories of Aging, and it's really worth it to support and buy this book. It's a great reference. It's very well annotated. There's a glossary, but I will refer to it in the future a lot. In particular, he takes to task some of these views of aging. One is the entropic view, which means that, to quote Chinua Achebe, things fall apart. One is a vitalist notion that you start off with a certain amount of life and then it erodes away. A nutritional theory of aging is also largely debunked. The genetic theory of aging, which is that it's in my genes, is also dissected. And then a very popular one these days with David Sinclair and Harvard and NAD is a free radical mitochondrial theory of aging. Again, he takes to task very logically those competing theories. His pet theory is the telomere theory of aging, and it's interesting that he knows all the people at the epicenter of this. The anecdotes are just so interesting, where he gets to spend time with Cal Harley, of course, the chief science officer of Geron, who patented TA65. Leonard Hayflick, the inventor of the Hayflick limit. Alexei Alovnikov. I mean, this is a gentleman, Michael, that has known all these men personally, and it adds a huge amount of credibility and texture to the history of telomerase activation. Third chapter is evolution, and it's a very, very brief chapter because Mike is not given to speculation and flights of fancy. He's more interested in what can be proved, and that's why I really appreciate his voice, because he is not of one world or the other. Specifically, he divides anti-aging into the enthusiasts, one might even say hobbyists, and then the serious people who won't even encourage the notion of anti-aging. And he likens evolution, the reason that we age and die, as a turning radius. In other words, the species can adapt quickly if you have shorter lifespans, and then reload different versions or iterations of the species. The fourth chapter is a search for immortality, and the funny analogy that comes up is where's the horse? When they first brought the first automobile into a repair shop, the guy kept asking, where's the horse? Well, the point is that most people don't assume that aging is something that can be prevented. And so this is an interesting chapter which goes over the whole history. If you want to know how these players all fit together, Noah Patton, Geron, uh, even the Nobel Prize winners, then you'll have more insight into there. Although the Cold Springs connection is not really elucidated, where Grider and Blasco and all these people, Harley worked. Fifth chapter, direct aging. He talks about immune, musculoskeletal, neurological, and cancer, and we'll discuss the excellent diagram that he came up with. And then sixth is indirect aging, or things that do not directly are not directly attributable to the cells, but let's say cardiovascular aging, the system of pumping against resistance, and Alzheimer's, which is actually his forte as a neuroscientist, and the fact that people have been treating the proteins associated, which accumulate, which would kind of be like treating the smoke and the soot from a fire rather than addressing the root causes of the disease. The penultimate chapter is slowing aging, and he gives finally some concrete recommendations. And we'll talk about the off-the-wall diet, which is basically the foods off the wall, the dairy, the fruits, the vegetables, not the stuff in the middle of the supermarket, which is processed and meant to store for months. Of course, he says you should eat fresh foods and pay the farmer and the grower, not the processor. Exercise, well known to slow aging, and comes out very strong in favor of telomerase activation. Finally, in the last chapter, he outlines IV telomerase therapy for the future and paints a very matter-of-fact picture of how we will all just go in every few years for routine maintenance and get buffed up. I just had my Rolex stop working after 15 years. I thought, boy, how unlucky. Susie's going to mail it off. And I talked to these Rolex guys, and they said, well, you're very lucky, actually, because you haven't had service for 15 years. So it was as though I'd driven my car for 150,000 miles and never gotten it serviced. So I guess it's all a matter of perspective. When we look at the 5 key analogies of the book, the first is the blind man and the elephant. And he says that when we look at aging, everyone has their own pet theories and research. And if the man is feeling the snout, he might think the elephant is a snake. If the man is feeling the tail, he might think it's a rope, and everything in between. And he says that, of course, everyone has their own expertise. That's the reductionist nature of science. But it perhaps is hindering us to not consider the whole elephant in the room, so to speak, which is that aging might be primarily cellular-based and even more so caused by telomeres, not just associated with telomere shortening. He likens it to cell phone contracts. If we have cell phone contracts that last only a few months, we'll always have fresh cell phones. But if they last 20 years, then all our cell phones will break. So it's always a balance of creative destruction, of renewing of the cells. As I mentioned before, he breaks people into 2 camps, the enthusiasts and the serious folks like the Nobel Prize winners who wouldn't even deign to comment on the idea of curing aging. And finally, he creates an analogy for diabetes. He says that they used to treat diabetes with bicarb, which is, of course, an antacid. But the reason that ketosis or acids build up in insulin is because of a lack of insulin and the metabolism that ensues. It's not going to help you to change the acid-base balance. You need to improve the insulin milieu. And that is the frustration he's seen with all these billions of dollars of research money for treating tau protein and amyloid plaques, which accumulate in Alzheimer's, because, again, we may be trying to use smoke evacuation and soot cleaner to prevent fires. Finally, he says, with the whole idea of curing aging, that they used to think in the early 20th century that treating polio would be a disaster. I mean, not only would it be a ruin for all the people in the iron lung industry, but you would only have children suffering for longer if you could cure polio. Again, being longer prisoners of their bodies. Well, what they are failing to see, what I hopefully and Michael are proselytizing, is that we're not talking about being old and infirmed. We're talking about looking and functioning as a young adult. That is our paradigm for anti-aging. It is not to have some ironic comeuppance where you live a long time but in a decrepit state. And unfortunately, people's cognitive dissonance won't allow them to even fathom that. This wonderful diagram that he put together shows what I think is a kind of a Rosetta Stone. If you recall, the Rosetta Stone was I think it was a sign from a bathhouse that was in Coptic hieroglyphics and Greek, and it allows us to decode the language. So what we're seeing here is that when you have telomeres that are long, and then you re-lengthen them, the cells are rejuvenated and they behave as such. If, however, you have telomere shortening, and I go through this in my book about how recombination occurs when the ends are excessively short, you get mutation and cancer, and that is represented there as genomic instability. What people don't talk about is that we may actually have gotten cancer many times and it peters itself out, it dies. These are not fully functional good cells. They are also prone to death just from replicative senescence, but other accrued mutations. So what happens if the telomeres continue to shorten, you can get enough poor function that the cancer cells die. And it's really an unanswered question whether we do get cancer hundreds, thousands, millions of times, and it just dies off or is treated by your immune system. So it's important to realize that, some of the obfuscating studies have shown that advanced cancers have long telomeres. Well, in my experience as a clinician measuring people's telomeres, I would concur that I think that people in the first year of telomerase activation therapy, if you want to call it that, they actually have a shortening of their telomeres because a lot of the abnormally long senescent cells die off. And so it's as though you finally got your first haircut in a Rastafarian. I mean, you get a median decrease of the length, but overall improve in the function. And I believe that may be due to apoptosis. So it's not all about length as long as it's not critically short. We talked a little bit about Margaret Chan, who's the World Health Organization director, who basically flew the white flag recently, said we should give up on a curative model of aging. This was almost 20 years ago. Carol Greider wrote to Mike, and he mentions in the book, he says, there's yet no evidence that telomeres in any way causes telomere shortening, that is, cellular senescence. Further, there's no direct link between cell aging and the aging of humans. Well, 20 years later, there are quite a lot of studies linking cell aging and different diseases, perhaps even aging of humans. So I wonder if she would retract that statement. In 2009, she and Jack Szostak and Liz Blackburn won the Nobel Prize in Physiology and Medicine for the discovery of telomerase. In fact, she was quoted in this year's Time magazine article saying, "Trying to boost telomerase through supplements is a very dangerous game to play, at least given the current state of medical knowledge." So these are our fearful leaders. One interesting thing that I'll be talking about soon is Carol Greider's discovery of perhaps a driver for the automobile of telomerase, if you will. And she has found that one of the tumor suppressor molecules, ATM kinase, is highly involved with regulating the activity level of telomerase. A week or two ago, we talked to Liz Parrish, who was the first woman or first person to inject herself with the telomerase gene with the point of increasing the gene copy number in her cells. Very interesting. She showed some signs that perhaps her telomerase activity was increased. When I read your book, there was something about your theory and my theory, if you will, that doesn't really jibe. And I talk a lot about the shortening of the telomeres in stem cells. And Liz and you don't really accentuate the importance of the stem cell type versus the somatic type, meaning like when we're taking telomerase activators or doing gene therapy, because the non-stem cells, if you will, are not telomerase capable, that they are less impacted. What do you think about the difference between, well, first of all, do you agree that somatic cells or more differentiated cells are less telomerase active?
In general, yes. I think the first thing I'd like to stress is that it's not really a black and white issue.
Mm-hmm.
You know, on the one extreme, you've got germ cells that clearly are capable of expressing telomerase. You have your typical somatic cells that, in general, don't express telomerase. But then in between there, when you're looking at stem cells, it depends what kind of stem cell you're looking at. Embryonic stem cell, hematopoietic stem cell, you know, and there are degrees of stemness, if I can call it that.
Yeah.
Likewise, there are degrees of the ability to express telomerase. And also, it's not a matter, even then, of 80%, 50%. It's also a matter of responsiveness. For example, if I look at hematopoietic stem cells, you find that they respond to certain cytokines. So you've got essentially a mother stem cell in your marrow that makes telomerase until it's turned on with a cytokine, then it does, then it's shut off again. Mm-hmm. So it's very complex.
Yeah, it is complex, which is why I simplified it. So in my book, do you think that it's just a red herring, that the longer the telomere, the younger the cellular function, and that there's something confounding that's accounting for that at a more fundamental level? Or do you think it's actually the length that signals the behavior?
Well, let me start by saying something peculiar, and doing it purely to create fuss. And I say it in my book, too. Is telomere length is entirely irrelevant. Yeah. Telomere length doesn't matter. What matters is change in telomere length. So if I have a cell, for example, say it's a specific variety of mouse and it has 150 kilobase pairs, that doesn't make it young. Mm-hmm. The question is, what's the pattern of gene expression? And in general, what you find is that telomere length per se is irrelevant, but changes in length sort of act as the conductor for the orchestra, and they overall set the pattern of gene expression. I like to say that we shouldn't be talking about telomere theory of aging. We should be talking about epigenetic theory or gene expression theory. Because what's going on at the key level of cancer initiation, at least as far as mutations go, is that you find that DNA repair goes down as the cell begins to act senescent. And the cell senescence is correlated with a change in telomere length, but not with absolute length. So if a mouse has 150 kilobase pairs, I still outlive it by 30 or 40 to 1. And yet it's the change that makes a difference. And maybe it goes down from 150 to 140 in aging, and mine goes down from, say, 15 to 10. It's equivalent aging, but I do it in 70 or 80 years, and it does it in 2. So again, it's not length. And even if you talk about the difference in length, the question still comes out, what's happening to the rate of DNA repair? You have to remember that all this is dynamic.
Yeah.
You know, you and I have had DNA damage occur since we started this conference here a few minutes ago, in probably every cell of our body. But in general, it's been repaired almost flawlessly. But if I take the 4 major types of DNA repair enzymes, localizing it, snipping it out, putting another one in and annealing it in place, all 4 of those sets of enzymes, their rate of replacement goes down with age, so the rate of DNA repair goes down with age, which increases the chance that at any given moment, I'm going to have a mutation that has not yet been repaired. Now, if I manage to go through a cell cycle with that, I'm a sitting duck for malignancy. And I think that's why if you look at the graphs, for example, of both humans and mice, the rate of cancer, malignancy goes up exponentially. And the curve matches whether you're looking at it over 2 years or 100 years. That is, humans over, say, 100 years and a mouse over 2 years. The shape of the curve is exactly the same. So clearly it's not a matter of time. It's a matter of DNA repair.
Yeah.
And we don't do it, and we do it very poorly as we get older.
And I thought some of your comments were really interesting for the book. Like when Cal Harley first was asked by you, he said only about 15% of aging is attributable to telomerase or telomeres. I wonder if he or Carol would retract that, given the thousands of studies now since they made those comments.
I think they both would. I think Carol Greider was right in 1996 to say that there wasn't a lot of data, and a lot of it was circumstantial. By 1999, that had changed. The comment I found much more interesting was that she said my book should have been banned. And that's kind of a compliment. The other remark I remember with Cal turned to me one day back in '96 and said, if he had a telomerase activator now that's 100% effective sitting at his desk, would I take it? And I said, no, I'd let you take it for a year or two, see how you do, and then I'll think about it.
Like, for example, you talk about these microglia, which are the immune cells of the brain, and they start to show dysfunction. The question is, first of all, do these immune cells, are they only created during a very early fetal embryonic development stage, or can they migrate in? And we know they can migrate in during times of inflammation, breakdown of the blood-brain barrier. But it's like sort of blaming the firemen for the fire in the first place. I mean, they're trying to clean up the inflammation and the cellular dysfunction that could be caused by the bad neurons and the astrocytes. What do you think? Are the microglia, are they originally there? You're born with the only ones you're going to have for brain immunity, or is it dynamic from the outside?
Let me say two things. One is, you're right, the literature on that is confusing and it's disputed. Are they migrants or are they native? Or are they some one, some the other? From a practical perspective, in trying to cure Alzheimer's, I don't care.
Hmm.
What I want to know is, again, where's the single most effective point of intervention? It's clearly not going after beta amyloid or tau tangles, because the success rate has been zero,
Yeah.
uniformly.
Intuitively, as clinicians, we're trying to cure people. And of course, that's a different focus from someone who's trying to develop a blockbuster drug or somebody who's trying to publish a lot and feed their grad students. So we both agree that there will be a cure and it will seem obvious someday, and that we need clinical trials of telomerase activating technology, and that people should look at the root causes. I just want to close with your last quote from the book, which is that we don't think this is going to lead to cataclysm or overpopulation, but actually it'll improve people, and that's been argued by greater minds than mine. And the way you end the book is by saying, to reverse aging is not merely to extend lifespan, but to extend the human spirit. To use this opportunity with elegance, grace, and compassion is to succeed at being human. So I think that really gets to the point of all these people that want to point fingers and talk about doomsday and quackery. They're missing the point. We're trying to keep people alive so we can become wiser and better. And I really applaud you, and I would urge everyone to invest in this book. It's a fantastic read and reference, and well worth it, in my opinion. So. But yeah, I think people, unfortunately, aren't able to conceptualize elephants. They just really, they get dogmatic about one argument. They say, well, mice don't age and they have long telomeres, or mice age and they have long telomeres, therefore everything you're saying and proposing is false. They'll get stuck at like, well, most cancers are telomerase active, therefore telomerase is bad. I mean, this is the level of discourse which people are able to engage in. Unfortunately, they're not able to look at systems, right?
That's exactly the problem. People are not looking at sophisticated, complex systems. They're looking at narrow little things. Now, sometimes it's just human nature. Example, if I've spent my entire life doing research on horses, nothing but horses and carriages, and you come along and say you've got a horseless carriage,
Right.
I'm against it. What you're doing is you're undercutting my entire life, and I don't like it. So again, if I've spent my entire life thinking mitochondria are the be-all and end-all of aging, and you say, well, I think it's more complex than that, there's a tendency to say you're obviously don't understand something.
Yeah.
None of us like to be told that you spent the last 70 years wasting your time. I wouldn't. I sort of don't blame them as human beings, but sorry, things are more complex than you think. Get over it.
The implications of whether Maria's Blasco's study in Spain and Rhonda Pino's study, whether they really showed that telomere re-elongation causes cell phenotypes to rejuvenate, is an incredibly important thing to understand. And yet people poo-poo it because it didn't get onto, the major meme pathway. I just find that baffling. Do you know
I
what I
know.
mean?
You know, I mean, we first showed that we could reverse aging. We first showed we could reverse aging in human cells now 16 years ago. And there, you know, there are two original problems. One is the paper was so hard to read that even when I knew the data and took the author out for beer, I really did. Um, it was hard to get the information out of the paper. But the other thing was part of what you're alluding to, which people don't believe it. The data's right in front of them, and they say, you know, I saw the car go down the road without the horse. There must be an invisible horse. No.
On your chapter, Dr. Fossil, on your chapter on slowing aging, you mentioned off-the-wall diet and telomerase activation. Can you elaborate on both of those as to how to go about those kinds of things?
Well, let me say this. Actually, I just was teaching that in a course the other day. You know, there are diets that clearly are going to make you sicker. One of my favorites was in 1890, there were German and American physiologists who were recommending sugar as nature's perfect food, which we regard as stupid. But I'm pretty sure we wait 100 years and people will look back at our recommendations and say, idiots. So there clearly are diets that will make you sicker, increase your risk of disease. And there are clearly diets that will lower your risk of disease, but I think there is no diet that will prevent aging. We know that caloric restriction, for example, may slow the rate of aging, but it doesn't stop it. It doesn't reverse it, certainly. There are such a things as good diets, and there are crazy diets, but there is no diet that's going to prevent aging. So slowing it, yes. Preventing it, no. I think we need to do something a little more interesting. And telomerase activators probably do that. They probably do slow aging.
And the types of telomerase activators that are available, I know TA65, Sierra has some things. What are...
Yeah, this is a real problem. You know, originally Noel Patton is in some ways my hero, because he's the one who took the Geron patent and ran with it as nutraceuticals. But the problem is it's sort of an unregulated industry, and that's good in a sense. I mean, I'm a anarchist from way back in many ways, very much a libertarian. But it also has problems, which is if I want to go out and buy a telomerase activator, I don't know much about the field. The odds are, I'll say to myself, that TA Science is probably as legit for a number of reasons. But what about the other guys that are charging half that amount or a tenth of that amount? And what if I just buy the root in Chinatown? And what if I import it from Thailand? You see what I mean? The problem is I don't know whether it's active or not. I don't know quite what I'm getting. And even at the best, I don't actually know what I'm getting. Now, I tend to trust Noel for various reasons. That doesn't mean I'm right. I know that. So it's hard for anybody who's going to buy this stuff to know what they're doing. I am pretty sure that if I go and I buy 40 milligrams of propranolol at the drugstore, it's probably propranolol. But if I go and buy astrologicides, a little tougher. And that's not even paying attention to, as I say, the issue of cost. It's not even paying attention to the issue of whether somebody's got a patent infringement. No, just trying to get it. Not clear. Hard. And then there's the other issue. Is it worth it? You know, patients ask me this all the time, and the answer is, well, it's a betting game. Telomerase activators probably, based on the data, probably have a role in preventing some or slowing the rate of age-related diseases. On the other hand, you know, if you're spending $200 or $400 a month on this, is that worth it? Well, if you've got $1 billion, it may well be. If you're on a fixed income and you've got $1,000 a month, then it is not, probably. But it's not an answerable question. It's a tough one. Actually, I am less than 65. I turn 65 next Wednesday.
Well, that's the provenance, the etymology of TA65 apparently is that Noel thought 65 was the retirement age back in the day.
Well, that's funny because here I am starting a whole new career. But keep an eye on Telesite. Let's see how we do. Right now, we've had a lot of people interested in investing in funding this, but we haven't had anybody signed up checks yet either. And as I say, we're moving ahead in our discussions with the FDA. We'll get there. It's time to get off my butt and do this.
Well, good. I'm glad that Liz inspired you.
Well, it's kind of
and you inspired her too. Yes.
Yeah. I had gotten involved with a company that had some internal problems, and I decided to do this anyway. And then Liz and Bill Andrews gave me a call and said they were interested. What about details? Could I tell them more? So I did. Now, Liz was more interested initially in FST than in using HTERT, but I said, try this. Here's why. So she's done it, and I wish her all the best.
Yes, I will report back to you how she's doing at the party
Well,
next.
she'll report back to me as soon as we get back in the country anyway. She's been out of the country for a bit, but we still text back and forth. She
Yeah,
says she's doing fine.
I think she's doing great. She has a lot of the soft signs of telomerase activation, like a very oppressive circadian rhythm and hypersomnia and vivid dreaming. How's the sculpting going? Did you give that up to write the book?
No, I didn't actually. I got paid to do it. Somebody paid me to do. I do art and sculpture, and mostly I'm a gardener.
Well, as you can see, Dr. Michael Fossil is a Renaissance man, a sculptor, a polyglot, and actually he's on a cellular and organism level reborn every day. So we look to him as an example. Hopefully he can continue to stay young and do these missions and really move products to market that are beneficial, especially in Alzheimer's, which is such a tragedy. But thank you so much for taking the time.
We hope it'll be a product, but more importantly, I'd like to get out there and cure people. You
Yeah,
know, if what you want to do is make money, there are tons of ways to do it. But I'd like to make this a better life for a lot of people whose lives are going straight downhill. Alzheimer's steals souls. I'd like to give them back.
absolutely. Well said. Okay, everybody, thank you so much for joining, and it's been a pleasure. This is Dr. Ed Park from Recharge Biomedical, and we will edit this and post it, and everyone have a safe and happy Thanksgiving.
Thank you all.
Thanks, Mike.
Bye, Ed.
Okay, take care.